Hi, yes, this is Michael Pino. I'm going to be presenting the investigation of novel antagonists for the Human Taar1 project. As we know, there are 6 different trace amine associated receptors within the human body that play significant physiological and neuropathological roles within the brain. We're going to concentrate on the Taar1, which is the most studied and conserved out of all the Taar family. And the Taar1 is also broadly expressed within the brain, as seen in this image here with the red dots located in like the prefrontal cortex, hypothalamus, and such. And also, the Taar1 is also a neuromodulator for the D2 receptor, as seen here. What is affected with the Taar1, whether agonist or antagonist, regulates what the D2 receptor does with the dopamine and gives us the various outputs that we want for the different studies that we are producing. This here is the workflow that we use in order to create the homology model, protein prep, various docking, MD simulation, and various virtual screening and analysis for this project. As we see here, the human and mouse TAR1 homology sequence indicated by the dark blue, it shows us that the human and mouse TAR1 is 74% similar to one another, while the light blue indicate that there are differences. And here is the ribbon representation for the human and mouse TAR1, with the human being the green ribbon and the mouse being the orange ribbon, with select differences in a few areas within the homology model. Since there's no crystal structure for the human and mouse TAR1, we had to create our own site map in order to identify the bonding pocket of the TAR1 receptor, as indicated here. And we use this to dock EPPTB, the antagonist for TAR1, that is the only known antagonist. And it shows that by the MMGBSA average, that EPPTB favors the mouse TAR1 system with an average of negative 106.7 versus the human TAR system, which the average is minus 96.5, which validates the Ki value of 0.9, which is for the mouse TAR, versus greater than 5,000 for the human TAR, which both of them validate EPPTB is good for the mouse and not the human TAR system. And once we ran the MD simulation on the human and TAR1 EPPTB complex, it gave us the 8 different clusters for the mouse and the human TAR1 EPPTB complex. And we took the human TAR1 complex here and ran a virtual screening with high throughput virtual screening here on the top 8 most abundant clusters, and we took the 16 most viable compounds that we saw within these clusters, and we did various analysis on them from docking scores, induced docking scores, MMGBSA, and within that, comparing the MMGBSA gave us 8 viable zinc compounds that looked feasible to look into, which this here is the protein ligand interaction diagram of the top 8 compounds, which shows us pi bonding, um, hydrogen bonding, and various other bondings to these zinc compounds. As we see here, there's 4 different residue contacts that are constant between all 8 zinc compounds that we have seen. And also with this, with the types of bonding, the 3 top or the hydrogen bonding, hydrophobic, and water bridging. In conclusion, the top 8 compounds seem to favor the Human Taar1 complex that we've seen. Most of the interactions of the top 8, like I said before, are the hydrogen bonding, hydrophobic, and water bridging within the interactions with the Human Taar1 structure. And the 4 residues that are constant between the top 8 are tryptophan, 264, serine 107, phenylalanine 199, and 195. Further investigation for this is assays to help validate the top 8 compounds, possible R group modification, and looking at blood-brain barrier permeability to see which one's better. Thank you so much for listening to my presentation.